recording, and we're officially in lecture mode. As always, interrupt in the chat or in person, or wave your hand, or do whatever you need to do to ask questions. All right. Today in our studio, we are on week 5, linear modeling. And really, this should be titled linear regression or linear regression modeling. We are not going to learn linear models and linear mixed effects models. For those of you who like to already do statistics, that is a different beast entirely, and it is a more complicated and also eventually going to be a more useful beast, but for now, we're not going to worry about that. If you are really, really interested in the statistical theory of linear regression and linear modeling, or you want to know more about how to apply more complicated models to more complicated data, there are 3 links here at the top of the page that'll get you going, and they're pretty good. I think the first 2 are actually, okay, so the first one is like a nerdy stats blog, and then the second 2 are actual tutorials written by graduate students at the University of Edinburgh, Scotland. It's a really cool program called Our Coding Club, and they are super great at walking you through like how to do everything step by step in a statistical program in R, or how to make a really nice graph. So if you're looking for tutorials, Our Coding Club, like OUR, is really a good place to start. So basic learning objectives for the linear modeling or linear regression tutorial here is we're going to learn the theory behind linear regression and linear models, which is an unbearably long-winded explanation of all sorts of statistical mumbo jumbo. At the end of that, you're going to get 1 line of code that you already have seen because I posted it in our figure code from last week. Oh, sorry, I'm not sharing my screen. Yeah. Here, why don't you check this out? There it is. There's the thing I've been talking about. Here's your links. Here's your learning objectives, and we're here. So I'm going to teach you the theory behind linear regression, which is great. And then I'm going to show you what a basic linear regression looks like and walk you through how to read the outputs. So you know what to ignore, basically, because there's a bunch of stuff that spits out after a linear regression that we don't care about. And then I'm going to try to talk to you a little bit about the limitations of linear regressions, and that's something that individual meeting people, we have done that a lot. We've talked about like when and when to not use a linear regression. And then I'm going to completely turn that over on its head and show you that you can, in fact, kind of force linear regression to do whatever you want. Don't get too confused about that. It's like not that big of a deal. At the end of the day, what I want you to know, really, is that you can run a linear regression to test a numerical relationship between a numerical variable on the x-axis and a numerical variable on the y-axis, right? So, for example, if you wanted to run statistics on any of our data that we got for the tide pools paper from figures 1 and 2 especially, you'll want to use a linear regression because we have a numerical value on the x and a numerical value on the y, and we're comparing the relationship between the two, or the ratio of x to y. In that situation, we need to use a linear regression and not an ANOVA. ANOVA is like group variables, and group variables are factors, right? So that's when you have groups on your x-axis. So when we use as.factor to make groups on our x-axis, where we have multiple y values for every value of x, we use an ANOVA. Otherwise, when we have numerical value on the x, numerical value on the y, and everything has a coordinate, like xy coordinate, every point has an xy coordinate, we want to do a linear regression or a linear model. Now, just to confuse you, and because it's true, unfortunately, you can, in fact, apply a linear regression when you have a group variable on the x-axis. I have told people over the last couple of weeks that like this is not the way I would do it. And it's not, but you can. And I'm going to show you that you can at the end of this tutorial, just because it's sort of enlightening to realize that ANOVA and linear regression are literally the same thing. They're the same, essentially. Cool. Feel free to jump in with questions or for clarification at any point, because I recognize that that last bit especially is a little bit mind-bending. I'm still trying to wrap my head around it myself. Honestly, I try not to think about it. If that helps, you can also do that. All right. So if you want to follow along with your own code, feel free. But I'm going to kind of zoom because this part's easy. We already know step 0. We want to check our working directory, make sure where we're at is the right place. So I did that and I'm in the right place. I have my R up behind this, so I'm happy to show you like line by line code if you want, but I'm just going to walk through the tutorial today because it's pretty straightforward. But if you want to see something, just like let me know and I'll do it for you. Okay, so we're going to load our packages. We want tidyverse because that is where we live and breathe, and it is our best friend. We want vegan, which is where we get our iris data because we want to use iris to plot in this case. We're going to use iris data. GGSci is just to make things look cool. Patchwork, again, is just to put graphs in the same, like multiple graphs on the same space. And then performance is how we check our model assumptions. So you'll remember performance from last week. That's where we get the function check underscore model, which is how we check every single assumption for our ANOVA. We can actually, all at once, with one line of code, we can use this exact same idea with our linear model and our linear regression. Check model from performance is maybe the most amazing thing that has ever been put into R. It saves so much time. Okay, so let's talk about what a linear regression is. First of all, according to statisticians and according to the stats blog that I posted at the top of this with a link, there are 3 types of data distributions. I feel like that's an oversimplification, but let me just try to talk to you about those. Each data distribution requires different statistical procedures. So when we have a normal distribution, which is our bell curve, right? That, or a Gaussian, if you're a statistician or a mathematician, which you're not, but if you like Gaussian, that's fine. I won't judge you. When you have a normal distribution, you can use an ANOVA or a t-test or a simple linear regression. They all can be used in these kinds of data. Now, there are 2 other kinds of data in the world, apparently. And I've only taught you the statistical approaches to deal with normal data, which means that the number 1 assumption for all of the statistical tests that you're learning in this class is that our data are normally distributed. If we have large enough sample size, we don't care about our assumptions. But if we look at our data and it looks super skewed and not normally distributed, we might want to consider transforming it. So if people run into that problem individually while you're doing your analyses, we can talk. But essentially what you want to do is you want to take your X or your Y variable and either square root it or raise it to a power or do a log or a natural log of it. And that will put it on a scale. If you do a logarithm especially, that'll put it on a scale where it's essentially from 0 to 1. The data distribution changes from like negative infinity to infinity to 0 to between 0 and 1, which almost always squishes it into something that looks like normal. Cool. And the way you do that is you just type log your column and it does it. Pretty straightforward. All right. So let's talk quickly about the 3 main types of data distribution. We already talked a little bit about the normal data. That is something that's called continuous data. We can use the statistical approaches that we know here. And this is when we're comparing traits like height, weight, nutrient content, and anything else you can measure across a centered, you can calculate a mean and an average. So if you can calculate a mean and an average, you can do our statistical tests, essentially. The other kinds of data are important just because we may see them in our analyses. We're still going to run the statistical test that I've taught you, just because they're the only ones we know, but recognize that they're not necessarily best practice. So this is something like count data. So count data is, say you're counting the number of individuals in a population, or you're counting the number of species within a community. So presence, absence data, that kind of thing, which we have done. We just didn't run stats on it. This happens to produce a lot of zeros, and in that case, you have to do a different kind of statistical analysis because the zeros are going to mess with your analyses. And then lastly, we have proportion data, which again is also 2 possible outcomes. So that would be like a presence absence, like a 1 or a 0. And in that case, we do want to run a different statistical test just because the zeros are going to like have an oversized effect on our analyses. feel free to forget everything I just told you. This is like not the most important stuff. If you're just like trying to get the basics, don't worry about this. But if you're interested in ecological data analysis, you will run into a point where you have a lot of ones and zeros and you need to do something different. And what you're going to want to do is something called a generalized linear model, which is a GLM, or a mixed effects model, GLMM. And there's a link at the top of this to like start learning how to do that. So like if you're doing a thesis, or if you're going into a master's program or whatever, or a PhD, this is a good jumping off point for ecological data analysis. So that's really all I'm telling you about it. If you're curious in exploring this and going into the field, like these are things you might want to try to look at. Okay. So when we're thinking about how to analyze our data, there's a lot of text here that I'm skipping. I just want to go back to this importantly. When we're thinking about how to test or how to analyze our data, we want to think about one thing first. What is our hypothesis? Our analysis and our visualization and our presentation of data, everything we do with data should be driven by a hypothesis. That is a good practice. When you're doing an experiment, you should have a hypothesis. That hypothesis could be the null hypothesis that I think nothing's going to happen. That's okay. But you should also have an alternative hypothesis, which is like, I suspect that there's this kind of relationship between X and Y, or between X, Y, and Z, because that is how you will think about how to like manage your data and run a statistical test. So I'm going to walk through how or like what a hypothesis might be for some data and how we would set up a test. All right. Before that, we need to talk a little bit about the theory behind linear regression. That was just kind of an intro to like data. A linear regression for most purposes, a simple one, where we're testing X and Y together, is essentially built to compare the correlation of one variable with another. So what we're trying to do in a linear regression is the null hypothesis for our linear regression, if we're looking at X and Y, is that there's no relationship between X and Y. Just like anybody's null hypothesis, we think nothing's going to happen. The alternative hypothesis for a linear regression is that there is a one-to-one relationship between X and Y, which is a 45-degree angle line going up or down, right? So as one grows, the other grows equally. As one grows, the other declines equally. Or as one declines, they both decline equally. So we're looking at that one-to-one 45-degree angle line as our alternative hypothesis. So if you're thinking about a numerical comparison between X and Y, linear regression is the one you want to use. That's basically all that means. Importantly, this is correlative. This is a correlation. If we see X growing and Y growing at the same speed, and we do a statistical test and show, yeah, there's a correlation between those growths, that doesn't tell us that there's a cause, right? We can't prove that X is driving Y or that Y is driving X. We can just prove that there's a relationship between X and Y, yeah? So there's a relationship between DO and pH, for example. That's what our graph shows us. We can see that correlation, we can measure it, we can put a number on it and say, it's this good. This correlation is 75% of the variation is explained by this relationship or whatever. Getting that causation, which is something that you're thinking about for your tide pool paper, like why, what's the mechanism behind DO's relationship with pH, is something that you need other data for. And you have it for the tide pool paper, so you need to like explore that. You can't say this graph shows me that oxygen drives pH or that CO2 drives pH. You have to, say that there's this correlation, and this correlation could be driven by, or is driven by this mechanistic process that we understand. So the graph shows correlation, not causation. You can prove causation through other means, but I want you to recognize that when you're writing your discussion. And I'm getting a lot of glare, so I'm going to close my window. Okay, great. On to the next. Linear regression has 4 assumptions. They're not very different from the ANOVA assumptions. One is we assume there's a relationship between X and Y that is approximately linear. No surprise there. 2, we think that our residuals are normally distributed. 3, we think that there's a homogeneity of variance, or homoscedasticity, which just means that the error or the distribution of data on the X and the distribution of data on the Y are approximately equal. And then lastly, we also assume that every data point is independent. We're taking independent samples. Again, independence, just like in ANOVA, is the hardest one to assess, and that is all based on experiment. And the rest of those, we can use our check model to check. And again, it's a judgment call as to whether you can run your linear model on your data or not. Okay, so we're going to check the following when we run our linear regression. First, we're going to look for nonlinearity. Like, is the relationship parabolic instead of linear? If so, we have to run a different kind of model. We're going to check for the homogeneity of variance of the homoscedasticity. And then we're also going to check for the presence of influential values or outliers in our data. And again, you can pull the extreme outliers out of your data if you want. We can do all of this with one line of code. We run check model, the name of our model, and then it spits out everything we need to see. All right, so that's one line of code that you learned so far, check model, and you already knew it. Now let's learn linear regression. A simple linear regression for the dataset iris. So let's take a look at iris. It's got sepal length, width, petal width, and then the species. That's it, very simple. So let's generate a hypothesis, and mine is going to be that sepal length will scale positively with sepal width. So I suspect there's a one-to-one relationship between sepal length and sepal width. As one grows, the other grows equally. So in this case, sepal length is my predictor variable, so it goes on the x. Sepal width is my outcome or dependent variable, so it goes on the y. And then I build my linear regression model, which is just LM with the notation y tilde x. This is just like the ANOVA. In fact, you'll see that this exact line is in our ANOVA dataset, or in our ANOVA tutorial. The only difference is we changed AOV to LM. So we changed from analysis of variance to linear regression model. That's it. Y tilde x data equals iris. And then we run summary just like we would for our AOV, and we get all sorts of nonsense. All right, so here's a quick rundown. Nobody cares about any of this. All we want is the adjusted R-squared value and the p-value. That's it. Our adjusted R-squared value is 0.007. So that the closer that value is to 1, the better our linear relationship is. So in this case, our linear relationship is terrible. The relationship between x and y is essentially not existent. There is not a linear relationship between x and y. And our p-value is not significant, suggesting the same thing. So we do not see a correlation between these two variables. Okay, if you want to learn how to interpret all of this other stuff, here is a nice little printout of how to do that. So the top portion here shows you just your model. So if you save this to an Excel document or something, you can see the type of model that you ran, just for comparison's sake. They totally skip these residuals because nobody cares about them. The coefficients are sometimes interesting, but in this case, they're not. They just show us the relationship of different variables. So just like in an ANOVA with a linear regression, you can add more than one variable to test the relationship. So I could have tested the relationship between sepal length, sepal width, petal length, and petal width altogether by using multiplication signs if I wanted. And then I would have gotten different coefficients for each one, which would have been interesting. That's not how we did it, though. Okay, so if we make the plot for that model that we just ran, the relationship between x and y, sepal length on the x, sepal width on the y, we can see that it's just a smattering of data. It doesn't look like there's a linear relationship here, right? Really, like, it just looks like a bunch of random points, and that is because it is just a bunch of random points. So strangely enough, even though sepal length and sepal width are two measurements of the same thing, the sepal, they are not correlated in a linear way. There maybe is a parabolic relationship here, like maybe, but at some point that becomes not important. Okay. So to visualize this with ggplot, as you've already learned probably from your code from the tide pools data, is you make your normal scatter plot, and then you just add one line of code, geom smooth method equals lm. And that tries to fit a linear model line to your data. And this method equals lm line is exactly the same information as this test that we just did with the lm above. Okay, so to add a geom smooth lm, is to put the line that approximates your statistical test on your graph. So the line is your linear regression model R squared value, essentially. And then the error cloud, the gray, is a confidence interval, a 95% confidence interval, which means that based on the modeling of our data, we can be 95% confident that all data would fall in this gray line if we were to pick a new data point. Okay, that is it for linear regression. So you learned check model, LM, and geom smooth. That's 3 lines of code. All right. So we ran all that. And like after we ran our model, we now want to quickly check our assumptions. So before we get too excited, are we actually able to run this test? Is it valid? So let's run through our check model. And remember, it tells us exactly what to look for. So non-normality of residuals and outliers, dots should be plotted along the line. They are. Moving on, that one's good. Non-normality of residuals, this should look like a bell curve. It does, so we're good. Homoscedasticity, dots should spread equally around the horizontal line. They mostly do. I'm going to consider that good enough. Here's another check for homogeneity of variance. They should spread equally. They kind of do, so I think we're good. And then check for influential observations. There's a couple of observations here that suggest that we may have a lot of zeros or a lot of really low numbers, but we don't have any other influential variables or numbers. So I'm just going to say that we're probably chilling. My official diagnosis is we might have some concerns about the homogeneity of variance, but it's mostly fine. Yeah, so I think we're okay. Essentially, if you don't get a Cook's distance value on the X for the influential observations that's greater than 1, you don't have a value to worry about. And I'm not going to really explain why. Just like this basically means we're fine, which is good enough for me. Okay, that is simple linear regression. I'm going to show you a harder one and a couple of examples further than that, but that is really all I want you to know. If you have a numerical variable on the X and a numerical variable on the Y, you use LM. You check your model, and then you add geom smooth to your plot. That's what you're going to do for your tide pool data. And this is a good statistical test to just check basic relationships between variables. Maybe you want to use it in your ecology near me or in your final project or whatever. Okay. Now I'm going to show you basically how ANOVA and linear models can be used in the same way. You can actually run a linear model on categorical variables. So here's MT cars, which is our friend that we've been running a lot of analyses with. And you can see that what I've done here is I've plotted MT cars by cylinder and horsepower. So cylinder on the X, horsepower on the Y, and I made cylinder into a factor. So now we're at the point where we're doing a linear model on a grouping on a factor on the X and a numerical variable on the Y. This is exactly what we use our ANOVA for. So just use ANOVA. But they are literally the same test. If I run my LM, HP by cylinder, I get a very similar thing to what I get in my ANOVA. And what it shows me is that there's an approximate linear regression or a linear relationship. The R squared value is 0.7, which if you're in a statistics class isn't very good. But if you're looking at ecological data, 0.7 is a pretty good R squared. So our approximation of our one-to-one line is pretty good. Our p-value is significant, suggesting that we do indeed have a significant correlation between X and Y. And then our p-values of our different coefficients, which the intercept one is equal to the 4 cylinders that you see right here. Cylinder 6 is right here, and cylinder 8 is right here. So that suggests to us that, in fact, the p-value for each one of those is significant, so that they're all likely different from each other, which is nice. So this estimate value here tells us what something that's called the effect size of each category relative to the intercept. So the mean of cylinders equals 4 for horsepower should be 82. That seems to check out. The mean for cylinders equals 36 is not 39, but it is in fact 82 plus 39. So 80 plus 40 is 120. So that looks like our mean. That's good. Cylinder 8, again, it's not 126. It's 126 plus 82. So that's 120 plus 82. That's like about 200. So that checks out pretty good. So our means have been approximated well with our statistical test. The p-values associated with each coefficient test the null hypothesis that each coefficient has no effect. This is all in the text, but I'm going to repeat this. When we're running factors in our linear regression model, the p-values test the null assumption that these different cylinder groupings have no effect. So there is, in fact, an effective cylinder, and there's an effect of each different kind of cylinder height size. The coefficient is likely to be meaningful, is basically all that means. All right. And then we can check our assumptions in the same exact way, and you can see that they're fine. Can you see how that's basically the same thing as an ANOVA, only a little bit more confusing? Just run an ANOVA. Don't do this. Just run an ANOVA. All right. Now, let's watch people's heads explode, shall we? Let's make this complicated. You can, in fact, make linear regressions more complicated, just like you can make an ANOVA more complicated through additive or multiplicative means by using a plus sign or a star or a multiplying sign. Here's Iris again. This time, why don't I hypothesize that there's an effective species on our linear relationship between petal width and petal length, right? So maybe each different species of iris has a different relationship between petal width and petal length. That's my hypothesis. So I graph our data. I do the exact same thing as I did before, but I'm adding this line for facet wrap, which actually makes a facet wrap tilde species will make a different graph for each species. That's something that we've seen in our ggplot tutorial. So I did that. You can see that the relationship does look a little bit different for each species. But it's super unclear, like, which one's the best and which one's the worst and like how different they actually are. So why don't we just not do the facet wrap, or why don't we add the linear regression model line using geom smooth to that same exact graph? It looks like this. So you can see that for versicolor, the species of iris in the middle, there's a really strong, very good looking linear regression line. For Setosa and Virginica, there is probably a positive relationship there, but it's not amazing. Doesn't look like a 45-degree angle line super well, and there's a huge amount of error. So let's plot all of those together on one graph by just taking out our facet wrap line. And you can see that the numbers for petal length and petal width are very different for each species. Whereas Setosa has the smallest petals, Virginica has the biggest petals, and versicolor is somewhere in the middle. But you can also see that the data for versicolor are very tightly wrapped around this line. So this is a really good relationship, whereas the other two are almost flat, which means that there's not a great relationship. So we pretty much already know what we're expecting when we run our linear regression model based on just visualizing it. But let's do it anyway so I can talk you through what it means. So here, we're testing the effect of petal width and species on petal length. And we want to test the effect of both of them together because we suspect that petal width and species together have an effect on petal length. If we wanted to test just the effect of petal length and just the effect of species separately, we would use a plus sign here instead of a multiplying sign. All right. So our linear model or our linear regression is the effect of petal width and species on petal length. Data equals iris. And we run our summary, and here's what we see. To start with, this is taking all of the data from here and fitting one linear regression across it. Okay? This one linear regression across all of our data has an R squared of 0.958, which is basically a one-to-one line, suggesting that, in fact, there is a nice linear relationship between petal width and petal length. But we can see that that varies probably by species. Yeah? So let's confirm that. Our p-value is obviously significant, suggesting that we have a significant linear correlation, positive linear correlation. Petal width increases as petal length increases, etc. Okay. Reading this coefficient table is a little bit tricky, but here's what it means. The intercept value is going to be the first species in alphabetical order, and that's it. So that is Setosa. So this is the value for Setosa. we can see that there is a significant effect of setosa, which is the blue, and there's a significant effect of virginica, which is the green. So these are different. The blue and the green are different from our one-to-one line in our plot, whereas versicolor, which is the one that has the nice linear relationship, is approximately the same. Okay, so that suggests that the relationship, the linear relationship between x and y for the blue species and the green species is different than it is for the red species. And we can see that it's not as much of a linear relationship. All right. What about the effect of petal width? Does petal width have an effect? It does not. And there is, in fact, a relationship between petal width and species at the same time, but it's only for versicolor, and that's not super important. But that does show us that they scale really well for versicolor. So overall, what we see, that species doesn't impact our linear regression model. Wait, whoa. Species does impact our linear model, sorry. And we can see a difference in slope between species if we look at petal width and species named together. So there's a difference in slope between versicolor, which is this one, and the slope of green and blue. But the slopes of green and blue are not different from one another. So we saw that, we've seen that basically twice now, right? So there's an effective species, and the effective species is that versicolor has a different linear relationship between x and y than green and blue, virginica and setosa. So we see overall a strong positive correlation between x and y, between petal length and petal width, and that species of iris has a significant effect of that correlation. So the slope of the line can differ by species. And we see that here when we're looking at our interactive term of species and petal width. And then we could check our assumptions, and they look fine, whatever. Assumptions basically always look fine. They don't look amazing, to be honest. There's a couple of wonky looking things, but for the most part, they're fine. Okay, that is about as complicated as a linear model gets for now. And I know that that's a little bit confusing, but that's all I've got in terms of linear models. Are there any questions on what we've done? Do you understand kind of where we're at? I'm going to show you what your homework assignment is, and then if you have additional questions about anything, please let me know. So your assignment is take any data you want, generate a null hypothesis for a relationship between two variables, and then assess the hypothesis with a linear regression. So you run an LM, show me a plot of your data, like a scatter plot or a box plot, and then a plot with the line added. So here you just do a regular ggplot scatter plot. Here you just add geom smooth. Then you do the summary of your LM and you describe the results. So I saw this kind of correlation, the p-value is significant, which means blah, blah, blah. And then you test your assumptions with check model. And then at the end, I just want you to say, like, we can accept or reject our null hypothesis, that there is no relationship. Or if you want to accept or reject an alternative hypothesis that there is a one-to-one relationship, that's fine. For me, those are the same. So that's really simple. You just pick two variables from any data set and plot them together and run a linear model. You can even use your tide pool data for that. You've got lots of examples to choose from. A little bit more complicated is I want you to test a relationship between two variables on the x and y and include an interactive term, so something categorical. So for example, like I did with petal length and petal width by species. So is there a difference between the relationship of petal width and petal length by species? I want you to pick any data set you want and mess with it in that way. You could, in fact, do this with your tide pool data if you wanted. You could test the relationship between, I don't know, DO and pH by tide pool, or DO and immersion time, or pH and immersion time by tide pool. That would be perfectly acceptable. And that's already data you've got your head wrapped around. So maybe try that. And like, if this goes off the rails, this too, that's okay. Number one, I'm expecting you to like actually understand all the way through and get right. Number two, I'm just expecting you to try to understand what's going on. And if you totally miss the boat on your understanding, I will leave you a comment, but I'm not going to take points off. Okay? Cool. Any final questions, thoughts, comments, anything you didn't understand that I can clear up? Okay. Well, that was linear regression, and that was your last R tutorial. So you are now armed with all of the skills you need to analyze all of the data for this class, which is cool. You will have 3 projects, including the tide pools, in which you are analyzing data in R. So you'll still get lots of R practice. Yeah, so if you're just here for the lecture, we're done. You're welcome to stick around and ask questions or hang out. Otherwise, it's office hour time. Thanks for coming, everybody.
Thank you.
I had a question about like when we would use an ANOVA. You mentioned it at the beginning of class. I just wanted to clarify. I took notes on it. So you said we use an ANOVA when we have a grouped variable on the x-axis. So if we had like, let's say we had like points scored by all the teams in the NFL, like by year, that would be an ANOVA case.
Yeah.
Okay.
Yeah, that would be a good use of ANOVA. For me personally, like I only really run a linear regression when I have a numerical variable on the x and a numerical variable on the y. So like I don't know. I'm trying to think of how to make this work for the NFL. Does like points scored in a game correlate with number of rushing yards in a game?
Yeah. Okay.
Which up until recently it probably did.
Yeah, but
people don't run the ball anymore.
Yeah.
Yeah. So yeah, something like that would be a good way to use a linear regression. And then if you wanted to see if that varied by team, you could use your linear regression and then add that interactive term of team.
Yeah.
Probably don't do that because that's like 32 different lines and that's going to be really confusing.
Yeah.
But you could. And yeah, for an ANOVA, anytime you have a grouping variable where you're going to have multiple y values for every value of x, you can technically choose linear regression or ANOVA, but it's so much easier to understand the ANOVA. Just run the ANOVA.
Yeah. Okay. Awesome. Thank you.
Yeah, sure. Good question. All right. Do you want to figure out your Tukey test?
Mm-hmm. Yep. And I guess the results of this also sort of goes into like the results that I got from, because I used the tide pool info to basically do this. Oh, I didn't